Hello students, good morning and welcome to my video where we will be continuing with our next segment. In our previous segment we have completed the protein, and in today's segment we will start with lipids, and after lipids we will be doing enzymes. Okay, so lipids is an organic substance, as we all know. Till now, whatever compounds we are studying, all are organic substance. Now lipid is an organic substance which are insoluble in water. Okay, insoluble in water, but they are soluble in solvents such as alcohol and ester. Okay, on this solvent these lipids are solubles, and the function of these lipids are they are used as the fuel reserve of the body. And they are the constituent of this cell membrane structure. If you remember, the cell membrane are made up of lipids bilayer, and these lipids are phospholipids. Okay, this is how cell membrane looks like. This is a cell, and this is the cell membrane. So cell membrane is actually made up of this lipid bilayers. So function of lipids, the constituent of cell membrane. And they are fat soluble. They act as a fat soluble vitamins. Some of the vitamins, they can get soluble in the lipids. Okay, they are soluble in the lipids. Okay, and along with the fat, these vitamins can be like how these lipids can be stored. Likewise, these vitamins also, along with this fat, they will be stored. Okay, so they are fat soluble. They help the vitamin to be soluble in the fats. Okay, fat soluble vitamin, and they are also a metabolic regulator. All the metabolic activities which takes place inside the cell. Okay, all the metabolic activities are regulated by the fats. They help in regulating, and then they act as an insulating material. They act as a insulating material. Insulating means keeping the heat, maintaining the heat in the body, not allowing the heat to pass out of the body. Okay, so having fats in the body, having lipids in the body, it helps to maintain the heat. It keeps the body warm. It maintains so that the heat does not. When we require this kind of condition, when the temperature outside in the environment is very cold, we need to maintain our body's temperature. For that, lipids helps to trap the heat inside the body. It does not allow the heat to move out of the body. Okay. Next, we'll see the types of lipids. There are three types of lipids: simple lipids, complex lipids, and derived lipids. Simple lipids, complex lipids, and derived lipids. So, simple lipids, you will see, they are the ester of fatty acids. They are the esters of fatty acids with alcohol. Okay, simple lipids are made up of fatty acids with alcohol. They are ester of fatty acids with alcohol. Okay, and example: simple lipids such as fats and waxes are simple lipids. Okay, true fats and waxes are simple lipids. Now, in true fats, we have example glycerol and fatty acids. Glycerol, and glycerol is made up of 3 carbon with 3 hydroxyl group with each carbon attached with hydrogen. So this is glycerol. And what is glycerol? It's a true fat example of a simple lipids, okay, which are made up of fatty acids and glycerol. Then we have fatty acids. As we are talking about, this fatty acid is also an example of true fats. They are, another example, fatty acids. They are made up of unbranched carbon atoms, unbranched carbon atoms, chain of carbon atoms with carboxylic group in one end. With carboxylic group in one end. Okay, and there are these fatty acids are of again 2 types: saturated fatty acid and unsaturated fatty acid. Okay, so out of this, saturated fatty acid is the waxes. Waxes are saturated fatty acids. The basic difference between saturated fatty acids and unsaturated is they solidify. Saturated fatty acid, they solidifies in the room temperature. Okay, like how you see wax, they are fat but they are in the solid form. And unsaturated fatty acids, they remain in a molten form in the room temperature. Molten form in room temperature means like vegetable oil. Okay, all this the other whatever the oil you find, and they are in the molten form. Okay, they don't solidify. Even if the temperature is low, they don't solidify. So some oil, they solidify in very low temperature, like your hair oil, you see coconut oil. When the room temperature is very low, then they solidifies. But in room temperature, the fats which remains in molten form, they are unsaturated fatty acids. Okay, so these are some basic. The next type of lipids we are seeing is complex lipids. So complex lipids are derivatives of simple lipids. They are derived from simple lipids. Derivative of simple lipids, along with additional groups. Okay, such as phospholipids. Phospholipids are lipids which has alcohol or phosphate groups along with it. Okay, lipids having alcohol and phosphate groups are known as phospholipids. Then we have glycolipids, in which lipids have additional groups such as glucose or galactose. Last one is lipoprotein. Lipids are associated with proteins, and proteins and lipids are associated, and such kind of lipids are known as lipoprotein. Okay, when glucose or galactose are attached with lipids, they are known as glucolipids, and when phosphate or alcohol are attached with lipids, it is known as phospholipids. So this is complex lipids. The last one we'll see is derived lipids. Derived lipids are again derivatives obtained on hydrolysis of simple and complex lipid. When complex lipids break down, okay, this means derivatives obtained on hydrolysis of simple and complex lipids means when complex and simple lipids breaks down. Hydrolysis means breaking down. Okay, when the compound, the bones are breaking down, that is called hydrolysis. So when the complex lipids and simple lipids they break down, they give some compounds, and those compounds are known as derived lipids. Example: steroids, terpenes, okay, prostaglandins. Now, simple lipids, we have seen glycerol's structure, right? So you can learn glycerol structure. Rest of the examples like phospholipids, steroids, derived lipids, and complex lipids, their structure you don't need to learn. Okay? Now we are done with the lipids here. So lipid is one of the biomolecules which we are studying in this chapter. Till now we have studied, if you can recall, we have studied nucleic acid, then protein, carbohydrate, and lipids. So we have completed all the biomolecules. Okay. So out of this, we will study the enzymes role and enzymes function, enzymes activity. So what is enzyme? Enzyme is a protein. I mean, if you see composition wise, what is enzyme made up of? It is a protein. Enzymes are protein. Okay. So we have studied protein, that they are made up of amino acid and they are polypeptide chain, and they play a various role. We have seen the function of protein, they play a various role. So out of that, one of the role is they play a role of enzyme. So out of all the proteins, we are going to study only enzyme in this chapter. Okay. So how do you define enzyme? A group of catalyst. A group of catalysts functioning in a biological system. A group of catalysts functioning in a biological system. Biological system means in our body. Okay, in our body system, inside our cell, a group of catalysts when they work. So what is catalyst? Catalysts are the substance which enhances the chemical reaction. Any reaction. So in our body, metabolic reactions are taking place. Okay, which are also a biochemical reaction. So these reactions are enhanced by catalyst. Okay, it doesn't mean a reaction cannot take place without the help of catalyst. Reaction can take place without the help of catalyst, but that reaction will be very slow. So what catalysts do? It enhances the rate of chemical reaction. It fastens the rate of chemical reaction. It helps to break down of the substances fast. It helps to make the substances fast. Okay. So enzymes are proteinaceous substance. And they enhance the rate of reaction. Okay. Enzymes are what? They are catalysts. Alright. Now, in our cell, these enzymes are found, and this enzyme will help in the reaction, metabolic reaction which takes place in the cell, inside the cell. Some takes place, some enzyme acts inside the cell, some enzymes moves out of the cell and then act. Okay, so we will see their types also. Catalyst. Now enzymes, when they are formed, when these enzymes are formed, they are in inactive form. Enzymes generally are in inactive form. So inactive form of the enzyme is known as proenzyme. Okay, proenzyme. Inactive form. Then they are converted into active form. They are converted into active form. Then they are known as enzyme. Active enzyme. Okay, how? How they are converted into active? Under the influence of specific factors. Under specific factors such as pH or substrate, etc. Like in our stomach, the proenzyme pepsinogen is secreted. Okay, pepsinogen is secreted. So when stomach starts secreting hydrochloric acid, the stomach's pH becomes low. Okay, it becomes 6.5 or 6. Okay, that is alkaline. Okay, acidic. So when the stomach's pH become alkaline, then this pepsinogen converts into pepsin, and that is when the pepsin start breaking down the protein. Okay, so if the pH does not change, okay, in the stomach, then pepsinogen remains pepsinogen only. And if pepsinogen, what is pepsinogen? It is the proenzyme, inactive. Okay, inactive enzyme. So can they break down the protein? No, they can't break down the protein. Okay, so that's why enzyme should be converted into active form to do the action. If they don't convert into active form, they cannot do the function. So how do they convert into active form? Either by pH or by substrate. Substrate means due to the presence of some other compound. Okay, substrates are some, it's like again a biological compound which are present inside the body. Okay. And then we see that enzymes are endoenzymes and exoenzyme. As I have told you, where does this enzyme formation takes place? Inside the cell. Okay, inside the cell, the protein synthesis takes place and enzymes are formed. Okay, if these enzymes act inside the cell only, then they are known as endoenzymes. Now, if these enzymes moves out of the cell and they act here, outside the body, outside the cell, any location of the body, maybe it may be intestine or it may be a buccal cavity. So if this enzyme moves out of the cell and acts outside the cell, if it acts outside the cell, so that kind of enzyme is known as exoenzyme. An endoenzyme which acts inside the cell. Okay? So next we'll see the properties of enzyme after which we will see the factors affecting the enzyme properties. So they acceleration initiate it. enzyme's function is to accelerate the reaction. Okay, they accelerate the reaction. They do not initiate the reaction. Reaction will be initiated by some other thing, some other factor. But once the reaction starts, then accelerate means what? Fastening up. Okay? So then this enzyme will fasten up the reaction. They don't initiate the reaction. So that is the first property. Second, once the reaction ends, at the end of the reaction, enzymes remain same. It do not change. They don't change. Okay? But the substrate will change into a product on which the enzyme is acting. That substance will change. Okay? But the enzyme itself will not change. Suppose this is A and enzyme E is reacting on A. So A will change into B. So after the B is formed, enzyme E remains E only. So A has changed into B, the reaction has happened, but enzyme does not change. Okay? That is the second property. And enzymes are larger in size comparing to the substrate. Substrate means if when I am saying a reaction, suppose A is reacting and converting to B. So A is a substrate and B is a product. Okay, and for this reaction enzyme E was required. Now this enzyme E, comparing to the substrate A, it is larger in size. Okay, it is larger in size. And when enzyme reacts with the substrate, now let me draw the structure of A converting into B. So what is A? A is a substrate, right? A is a substrate here. So let me draw the enzyme's structure. So how enzyme structures are? Suppose I'm drawing an enzyme here. So enzyme has a site here where this substrate A, and where is A? Now this is A and this is the enzyme. Okay, so this A, how is the structure of this A substrate? It can fit here. A is like this, that it can fit in here. Right? So when it fits here, then enzyme will act upon this A, and then this A will convert into a product. What product? It will convert into product B. Okay, it will become product B after the action of enzyme. We will see that enzyme action we will see later. I'm just trying to make you understand what here the structural part of E is. Now the part where the A gets fit in here, so that is called active site. Okay, so enzyme has an active site where the substrate A is a substrate. A substrate comes in contact with the E. So this contact site is known as active site. Okay? And then enzymes are amphoteric in nature. Amphoteric nature means they can react in acidic and they can react with acidic and alkaline substance both. They can react with acidic and alkaline substance both. Okay? And enzymes shows specificity. Specificity means single enzyme acts upon a single substrate. One enzyme is meant for one substrate only. That enzyme will not act upon other substrate. Okay? one enzyme will act only in one substrate. So that is known as specificity. Okay. For example, urease is an enzyme, name of an enzyme. So it acts on urea invertase. So they don't act upon any other substance, but they act upon only one. So this single enzyme will act upon single substrate. For example, salivary amylase will act upon only starch. Okay, they act upon only starch. Amylase cannot digest protein; it can digest only carbohydrate. Okay, so they are specific. They are very specific with the action. Alright, so here we come. So I conclude the video here today. So in next class we will complete the enzymes portion. Okay, factors affecting and the enzyme action, enzyme reaction. Okay, and nomenclature of the enzymes. All these portions which are left we will complete in the next video, next segment. Okay, thank you, and read, go through it thoroughly, see the video thoroughly, and then if you have any doubt, please let me know. Thank you and have a good day.